Okay, so we talked about the first two parts of our model for seeing color. The third one is our observer. Mathematically described by the spectral response curve. The curve describes the observer in terms of the amount of light energy that is detected at each wavelength. So the first curve was how much light is produced at a wavelength. The second curve was how much is reflected. Now this curve is how much is detected by the observer. The higher the graph, the more light of that wavelength is detected, just like all the other ones. The most common observer of color is the human eye. We all have one of those. And the picture is as follows. Got to have a picture of an eye in there. Light comes in through here and is focused on the retina. We had this stuff last time. The fovea is a part of the eye where what is, remember? What's in the fovea, remember from last week? Cones, right. And cones are sensitive to color, right. Rods, which are the other one, are dispersed throughout the retina. And those are the ones that see just intensity of light, don't detect color. So we have two types of sensors. So it kind of works like a camera where the light is focused in there. If your cornea here is out of shape, then you need glasses, like some people need glasses. And if you have surgery, sometimes they'll fix the shape of your cornea, I believe. So consider the eye as a camera through which we observe the world. It contains an autofocus lens and a variable aperture called the iris to control the amount of light. So, you know, when our pupil gets big in the dark, we can see better in the dark. When it's real bright outside, the pupil looks really small because it doesn't need as many of those sensors. Lens projects an image of whatever we look at onto the retina, which covers the back of our eye. And the retina is sensitive to light just like a film in a regular camera. It's far more sophisticated, as you will soon discover. First type of detector are rods, and they're called that because of their shape. Very sensitive to low levels of light, allow us to see in a dark room, and they'll shut down in bright sunlight. That's why when we walk into a dark room after being in the sunlight, not many of our detectors were working, so it has to reestablish itself so we can see. And the other type of sensor are called cones. They are our color sensors, and we have three types of those. They are extremely sensitive light. That's what give us night vision, and they can be overloaded, like I mentioned before, and turn off. Cones are located almost entirely in the area of the retina called the fovea, although there's some. The interesting part is that there are about 7 million of them packed into that small area. But they are able to distinguish between different color wavelengths. We've talked about the different color wavelength earlier today. Our our cones are very sensitive to those individual wavelengths. And some are sensitive to certain wavelengths, and others are sensitive to other wavelengths. One type is sensitive in the blue region, one in the green region, and then one in the red region. So generally we consider to have blue, green, and red sensors in our eyes. That's how the curves look. So one is blue, one is green, and one is red, although the colors are a little goofy there. Note that the response of the three types of cones corresponds to the light primary colors. Last week we had talked about that, and that's why we consider blue, green, and red to be the light primaries. The fovea is located in the center of vision so that when you look directly at an object, the image of that object falls directly on the fovea so we can see its color. You'll see in a later class that when they first did a bunch of studies to determine what's called the standard observer, they looked at a very small area where most of that light fell on the fovea and not the rest of the retina. And then in later experiments, they did a larger area that the light fell on more of the retina. And it turns out that the calculations for that second one are much closer to we actually see, or how we actually see colors. So it's more accurate. We'll talk about that next class, I think. Very high concentration of cones in the fovea gives us great visual acuity, the ability to see extremely small details. These are additive primaries. That's supposed to be white there. I've tried like 3 or 4 times to get it to come out white. Didn't. We have our optic nerve system that connects basically our eyes with our brains. And we talked about the blind spot last time. We don't normally notice because, again, our brain just filters that out. But that's kind of the cord that connects the two of them. There's hundreds of millions of nerve cells which are interconnected in an extremely complex fashion. Don't really know how it works, but there's some combining of the signals from our eyes that happen in there so that when the signal goes to our brain, goes in 3 signals, if you want. We have, and when we talk about color, we'll talk about this in terms of our LAB system, that we have a signal that represents lightness and darkness, white and black, and then we have a signal that represents yellow and blue, and a signal that represents green and red. So we can make a graph, plot, we call LAB space, that we'll talk about a lot in this class. And the idea is that the green, red, and the blue, yellow are kind of a continuum. You can be either yellow or blue, but you can't be yellowish blue. So you can start up here, go towards the center being gray with basically no chromatic color to it, and then you go on the opposite side, you start getting bluer. Same with the green and the red. So that's kind of how our signals work. You have a red-green scale, a blue-yellow scale, and then a lightness-darkness scale. And that's the way we'll be picturing colors a lot once we are looking at colors more specifically. The following curves represent 3 detectors used for seeing colors: the human eye, and then 2 detectors that are used in spectrophotometers. One's called a photomultiplier tube, and one's called a silicone photodiode. And what you see here is that the 2 artificial ones have much different responses than our eye really does, which is okay because we can mathematically adjust it. The important thing is that they all have response between 400 and 700. But again, you can see we have to use, when we use artificial means, it doesn't work exactly like our eye does, so there's some compromises with how that works. The next slide shows how the curves of our model for color are added together to result in a stimulus for color in the human brain. We will perform a simpler but similar calculation as an exercise. Be based on the slide after the next. So basically, how we calculate our stimulus for color is we have our light source and our object, and we multiply those together. We get this curve. If we multiply this curve by each of the individual curves that represent the sensitivity of each type of cone, we multiply this curve times that, and we get that. This curve times that, and we get that. That curve times that, and we get that. And we have our 3 components of our stimulus for color. We'll do one that's a little simpler than this when we actually do our calculations. But if you ever wondered how or what you would use calculus for, this is one example. You can find the area under this curve using calculus. So that'll probably make you want to jump out and learn how to do calculus, I'm sure. But as we get into the next chapter, we'll talk about this a little bit more specifically. Our a little simpler model is this, where we have our spectral power distribution curve. We multiply it times our reflectance curve times our response curve, and then we'll get a single curve for our stimulus for color. We can describe colors in a different way using names instead of physics of light and materials. The question is, how do we organize colors into a rational system? Generally speaking, we use the terms hue, lightness, and chroma to describe the 3-dimensional color system because we have to use 3 dimensions. So we have, if we make a model of it, it'll be in 3-dimensional space, just not on a flat surface like the board. First one is hue. That's that attribute of color which makes it appear red, yellow, green, or blue. So, the hue of something is red, it's blue, it's yellow, Not sure how else to define it other than that. The hue. Chroma is the attribute of a color which describes its purity in relation to gray. The closer to gray the color is, the less chromatic it is. So think of a real deep, bright color. That would be very chromatic. If it's a dull gray color, that would be less chromatic, or low chroma. Color's distance from gray increases, the color becomes more chromatic and vivid. Lightness is the attribute of color which defines its relative position on a continuous scale running from black to white. A pastel green is closer appearance to white, while a deep green is closer to black. So the pastel green is lighter, the deep green is darker. And we can describe any color that we see in terms of its hue, its chroma, and its lightness. And we'll actually do it. That Munsell book that you have there has some exercises in looking at colors in terms of those three. In your book, there's a couple descriptions of experiments, the desert island experiment and the art classroom experiment. I'll let you read those. And then there's some charts in there that show how color can be organized in terms of different attributes. We'll get into this much more in the next chapter. And here's another coordinate system. We'll use this type of system where lightness and darkness, black, white, go up and down. The hue goes in a circle around that line going up and down. We'll talk about the hue circle. And then the chroma, the very center of this cylinder, if you think of it as a cylinder, is achromatic, or just grays, lights, darks, whites, blacks. And then the farther out you get from the center, the more chromatic the color is, the more red it is, the more blue it is, the deeper the color. Just another type system there. There's some other terms they talk about in the book. Chromatic adaptation, changes in the visual system that approximately compensate for changes in the spectral quality of illumination. So if you slowly change the light source, for instance, it'll appear to you that the color of the object doesn't change. But if you did it quickly, then you could see the difference. So our eyes and mainly our brain have a way of adapting to what should be there more so than what's actually there. And then light adaptation, changes in the visual system that approximately compensate for changes in the level of illumination. So as the light goes up or down, if it does it slowly, we'll perceive the object the same way as it was when the light was lighter. We just kind of compensate for that. Our brain says that's what it should be, that's what it's going to be, don't care what's actually there. You know people like that about a lot of things, right? Don't care what's there, I know what the right answer is, and don't confuse me with the facts, I know what's right. Color consistency, the general tendency of a color of an object to remain constant when the level and color of illuminant are changed, similar to what we said before. So those are just some definitions there. And the last one is a real important one that we run into in color all the time. Phenomenon in which the spectrally different stimulus match to a given observer. Two objects match under one viewing condition but do not match under another condition. Mainly that happens when we have two objects match under one light source, we change the light source, they don't match anymore. That can be a big problem if you're matching a color and say you match it under daylight and then you send it to your customer and he looks at it under a fluorescent light and says, hey, that doesn't even come close to matching. That can happen. So two specimens match under a specific illumination to a specific observer and whose spectral reflectance transmission differ in visible wavelengths. That's a definition according to ASTM for metameric match, also called a conditional match. It only matches under certain conditions. And this is another one that's in your book that probably will look better. But these are two different grays that have two different curves. And it shows this one has more of the straight curve, that one is more of the curvy curve. And under some conditions of light, those two colors will look the same, but in other light sources, they'll look different. That's what we define as metamerism. The only way you can have no metamerism, no matter what the light source, is if the two curves for the two materials are exactly the same. And the only way you can have that is if you use the same colorants to make it. So one thing that you could tell by looking at these two curves that you couldn't necessarily tell by looking at the colors is that this formula uses a certain set of colorants. This other formula uses a different set of colorants. But under some conditions, you can get both of those formulas to look the same. But not under all conditions, only under very specific conditions.